Okay, so last time we talked about the lipids and we talked about the triglycerides. Triglycerides is one glycerol and three fatty acids. It's the same way that we did when we talked about the synthesis of carbohydrates. Basically, it's called dehydration synthesis versus hydrolysis is the opposite. So this is dehydration synthesis. Look at this one here. This is glycerol up here, the purple one or the move one. And you're going to take an H from this one, an OH from the fatty acid. You add it. This is mono glyceride. You add another one diglyceride. You add another one triglyceride. And look at the triglyceride here. You ended up getting three waters because this is what dehydration synthesis. Different types of lipids. We have steroids. I talked about this. And basically, it's a four-ring carbon structure, four rings. You don't need to know any details with these simples or reactions and all of this, but just know that it is four-ring. Examples, cholesterol, sex hormones like estrogen in females, testosterone in males, steroids, if you heard about the corticosteroids, or if you heard about cortisone, it is one of them. Bile salts are also the same. This is the four-ring structure. You don't need to know any details. Just know it's four structures, four-ring structures. You can add things to the lipids for a reason because we talked about lipids before, and I mentioned that lipids are hydrophobic, right? Phobic. It does not like water. It does not react with water. It doesn't mix with water. So in order to make lipid, a special type of lipid that at least part of it can interact with water or part of it can be hydrophilic, you can either add a phosphate group and you call it phospholipid, or you can add sugar and you call it glycolipid. Glyco is a prefix that means sugar or carbohydrate. We're learning prefixes and suffixes as we go. Glyco, it's carbohydrates. So you can add carbohydrate to one end, or you can add phosphate to one end, so that this end is going to be hydrophilic. And there is a reason for that. You will learn more in the next chapter when we talk about the membranes, that we have to have one end that is hydrophilic. So look at this. This is the head, and this is the tail. This is the main structure right here. This is glycerol. And how many fatty acids do you see here? One, two. You see those two? One glycerol and two fatty acids. What do you call it? Diglyceride. Diglyceride. So this is diglyceride. Add phosphate to it, phospholipids. Add sugar to it, glycolipids. Did you get the idea? So this end that have the phosphate, or this end that have the sugar, this end is hydrophilic, which is, we call it the head. Look at the head. Now look at the tail right here. This is a fatty acid, nothing added to it, so it's hydrophobic. So you end up having this structure, head hydrophilic, and the tail is hydrophobic. There is a reason for that. We will discuss it. Look at the glycolipids. Obviously, down here, this is the diglyceride. This one glycerol and two fatty acids. So this is the tail that's hydrophobic, and the head is hydrophilic, so that you end up having this, the membrane. You will end up having, we will discuss it in the cells again, but you want the cells to have a head, the membrane of the cells to have heads that are hydrophilic, and the tails are hydrophobic, as you see here. Hydrophilic out there and hydrophobic here, or this is the phospholipid, the orange. Look at this spherical structure here. This is the phosphate, which is hydrophilic. Why? Because you're dealing with water, and the tail is not dealing with water. Proteins, also one of the organic molecules. It goes without saying. All organic molecules, carbon and hydrogen, right? But the four that we're doing right now contain also oxygen. So all four contain what? Carbon, hydrogen, oxygen. What's the difference then? For the carbohydrates, I mentioned that the ratio is 1 to 2 to 1, right? For the fat, I mentioned there is a carboxyl group, COOH, and at one end. Here, there is a big difference, that's nitrogen. Carbohydrate and fat, no nitrogen, zero nitrogen. Now we have this nitrogen. So even though you don't need to know the structure, but if you look at one end, you see COOH. This can be fat or protein. How to know them from each other? It's a bunch of carbohydrates, a bunch of carbon, hydrogen, and oxygen. How to know what it is? One end have COOH. This is not carbohydrate. Okay? It is either fat or lipids or proteins. How to know those from each other? Both of them have an end that contains COOH or carboxyl group. Yes, look at the other end. If it contains nitrogen, done. It's protein. Other than that, will be too much. Don't worry about it. I will show you. How many, or what is the building unit, or we call it the monomer of protein? Amino acid. The polymer is protein. The monomer is amino acid. So I don't have to say what is the building unit. It's the monomer. Same thing. So like if I ask you what is the monomer of carbohydrates, monosaccharides. You guys remember this? Glucose, fructose, and galactose, right? What is the monomer of lipids? Fatty acid. What's the monomer of proteins? Amino acid is a monomer. And we will see the nucleic acid, it's going to be nucleotide. So there is a building unit that we call it monomer for each one of those, and you have to know it. We have 20 different types of amino acids. We put them in different ways to make proteins. Generally speaking, the functions of proteins in our body, and this is important to know. What are the functions? Very important. Part of it is structure inside the cells and inside the muscles and inside a lot of structures. It is for support. In our muscles specifically, this protein is for movement. Transport. When we do the blood, you will see that there are carriers in the blood that are protein in nature. So it's transport. And part of the proteins are also buffer. Buffer means regulate the pH, and we talked about this. Do you remember? Weak acid, weak base. Enzymes, hormones, and antibodies. All these three are protein in nature, and this slide is important. Enzymes, hormones, antibodies, all these three, what's the chemical nature? Protein. So in order to make the protein structure, you have to get a long chain of amino acids. It's different levels, okay? The first level is like, how can we build this whole building here, H, or J, I mean. You start from here, from the very, very beginning. I get bricks, put them beside each other. You end up making a wall, right? You bend the wall, you're making a room. Finish the whole room. This is the three-dimensional structure, which is one room, right? Is that the whole building? Not yet. Make many rooms. That will give us the whole building. And that's exactly the idea of building the protein. You start from long chain of bricks, or in this one, long chain of amino acids. And each amino acid, I will show you in the picture, will have, look at this end, carboxyl group, right? One end will be carboxyl group, but the fatty acid also have a carboxyl group. Yes, look at the other end. Look at this amino group, NH2. It is not fat, for sure. I will show you the picture now. There is a central carbon, there is hydrogen, and there is something called R chain, which is a side chain. So look at this structure right here. This is the structure of the amino acids, central carbon, hydrogen here, side chain here, but look at the ends. This one end is carboxyl group, which is the same as lipids, but look at this end, completely different. NH2, amino group. So now, how can I make a chain of amino acids? You get one amino acid, you get another amino acid. Do you remember the dehydration synthesis? Dehydration synthesis. Here is one amino acid, one amino acid. I want to link them together. I want to make a bond. You can't. You have to take H from one, OH from the other one, then they're gonna make a bond, right? Do you remember these? So this is called dehydration synthesis, obviously, right? So I get one amino acid, dehydration synthesis, they attach to each other. One more, another one, another one, another one. But the one thing that's unique here is this bond between each two amino acid has its own name. It's called peptide bond. Peptide bond. So this is something unique here. Peptide bond. So since it's amino acid, bond. Another amino acid, bond, 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 bond. I mean peptide bond, peptide bond, peptide bond. So together it's called polypeptide. Yes, you have a question? I'm sorry? Yes, exactly, yes. So you end up getting what? Polypeptide, several peptide bonds, right? So now what do I have? Amino acid, amino acid, amino acid, amino acid attached to each other by peptide bonds. So what is this? What do I have? Polypeptide. Do you mean protein? Not yet. Too early. It's just a chain of amino acid called polypeptide. Okay? So look at this. Of course, again, you don't have to know the details. I'm just telling you that this is amino acid, this is amino acid. Remove H and OH from each one of those. Put them together. They will link, get the hydrogen. What do you call this? Dehydration synthesis. I repeated several times. So we call this dehydration synthesis. And what's the type of bond that you made here? In a protein specifically, you have to give it its own name, peptide bonds. So you made a peptide bond. Can I split them back? Yes, it's the exact same thing. And what do we call this process? Hydrolysis, which is the opposite of dehydration synthesis. So the first part that I talked about is called the primary structure. Long chain of amino acids. I'm making a peptide, a polypeptide. What do you call this process or this step? Primary structure. It's just a long chain of polypeptide, a long chain of amino acids. That's it. Like you're making a wall now. Is that a building yet? Not yet. Just a wall. So this is primary structure. Secondary structure, you are going to bend the wall. See this? The wall goes like this, and then you bend it, and then you bend it, and then you bend it, and then you put the roof. You're making a room, right? So this just bending is the secondary structure. It will look different. It's not just a straight chain like this. No, you go to like bending, bending. When you bend it, this is called the secondary structure. And in order to keep it bended like this, and it doesn't go backwards, you have to keep it like this. You have to put hydrogen bonds to keep it in that shape. You guys remember the hydrogen bond? The weakest type of bond? We talked about this. So that will give you the secondary structure. I will show you the picture. Then what? Tertiary structure. I'm going to make this, instead of just bending, I'm going to make a whole room, three-dimensional. So this is called the tertiary structure, like this room. This is the tertiary structure, three-dimensional. Everything is put together. Quaternary structure, quad, similar to quad, four, right? So four rooms. You put four rooms together. Here is one bedroom, another bedroom, kitchen, bathroom. This is a house, which is a protein. So look at this picture right here. This is the primary structure. Can you see this? It's just a long chain of amino acids. That's it. It's a polypeptide, linear. Now we are going to bend it. Look at this. This is one form of bending. You make it twisting like this, like a spiral. And what keep it in that shape? Hydrogen bond. So now I got the secondary structure. Or you can make it like this. It doesn't have to be twisted like this or spirals like this. It can be pleated sheet. This is called pleated sheet or beta sheet. Okay? So I can do it like this, spiral, or I can just do it bending like this, like zigzag. This is called beta sheet. And again, to keep it like this, you have to put the hydrogen bond. Now, how about the tertiary? The tertiary is three-dimensional structure, not just bending, a whole room, three-dimensional structure. And then here is a three-dimensional structure, tertiary, like a room, another one, another one, another one. Put four rooms together, you get the full house, which is the protein itself, the quaternary structure. Quad is four. So look at this. One, two, three, four. Two blue and two purple. This is the globin, which is a protein, part of hemoglobin. I think we know hemoglobin, right? Hemoglobin in the blood. Yes, hemoglobin is globin, which is protein, of the red blood cells. If you get one red blood cell and open it, what do you have inside? Hemoglobin. What's a hemoglobin? It's globin, which is a protein. One, two, three, four. You have four proteins here. Plus, you add other things to it, not our issue now, but just hemoglobin is a form of quaternary protein. Here is another quaternary protein, collagen. I'm not asking you to know the details, just examples. Here is hemoglobin, quaternary. Collagen. Do we know the collagen in our skin? You have, like they say, take this shampoo with collagen, it will make your skin better and stuff like this. So collagen in the skin and hair. Collagen is also protein. Some proteins will be globular proteins and others will be fibrous proteins. So globular proteins is like a spherical structure. Look, globular structure. It looks like a spherical shape, versus the fibrous one, and this is looks like a sheet, like the collagen one. So these are just different shapes. Enzymes. Remember, I already mentioned hormones, enzymes, antibodies. All of these are proteins, right? These are different types of proteins. Enzymes specifically, I already talked about it before. If you guys still remember, I told you if your car got a flat tire and if you don't have good tools, it will take you effort and time, right? If you have just a couple of wrenches or something, it will take you 3, 4 hours to change the flat tire. But if you have this hydraulic machine, it saves you time and effort, isn't it? This hydraulic machine is similar to the enzyme. So what's an enzyme? Enzyme is a catalyst of the reaction. Speed it up and use less energy. Um, how does the enzyme know that this is my reaction? How do you know that here is sodium and here is chlorine and there is a reaction here? How do I know that this is my reaction that I need to catalyze or I need to help? I need to speed up. How do I know? Because there is an active site. There is an active site in the enzyme. If you fit, it is yours. Okay? Enzymes will have certain criteria that you have to know. Specificity is one. Not any enzyme help any reaction. This enzyme help this only. Okay, this is sodium and chlorine. There is an enzyme for this, an enzyme for something else. Each reaction have its own enzyme, and we call it is specific. Each enzyme is have a specific substrates to work on, so we call it specificity. These three are important criteria that you need to know. I'm trying to finish fast, but when you see me focusing on something like this, obviously it's important. Criteria of enzymes, what does it do, and the criteria. Specificity is one. Saturation limit, you cannot make it like do tons of work. It will be saturated. Regulation, you can tell the enzyme what to do. It's regulated. So basically like this. If you ask me like carry one chair, okay, I can do it. Carry two, okay. Carry 15, no, I can't. I have my limit, right? So it can do a limited amount of work. We call it saturation. If you give me like 5 chairs to carry, this is the maximum. You give me one more like the weight builders or the weight lifters, they give them like more, and I can do like 100, 150. You go to the maximum. If you give him more than that, it will going to fall, right? You reach your limit, saturation. Regulation is, can you do two? Yes. You know what? We are in a hurry. Make it four. So you tell them what to do. Regulation. So if you look at this, this is the enzyme. Look at the active site. Only this S1 can fit in this, and only this S2 can fit in this. So every enzyme has active sites that have a specific shape. If you fit, you are mine, I'm going to help you. You don't fit, you're not mine. So this is how the enzymes know, which is specificity. This enzyme can work on if you have S1 and S2 reacting together, I can help you. Any other enzymes? No, will not be able to help. Can this enzyme help anything else? No. I can only help those two because you fit this here. Look at the shape. And you fit this here. Fit, come together. Okay, I'm going to help you. So this is specificity. Look at this, the fit. So you make the reaction, you help them catalyze the reaction, and you end up having the product at the end. Here is the product leaving. So now the enzyme can help the next. If you ask the enzyme to do this work continuously, it's okay. But if you go beyond its capacity while still working on something, no, you reach the saturation limits. Not only that we need enzymes to help the reactions, make it faster and less energy, but we need also some other things to help the reactions. This is called cofactors and coenzyme. Co, CO, is a prefix that means together. Okay, so cofactor is a factor that goes together with the enzyme to help the enzyme start. Cofactor. Coenzyme is also a cofactor, but it's non-protein organic. Non-protein organic specifically. Anything else can be a cofactor. Okay, iron can be a cofactor, just iron. Magnesium can be a cofactor. A lot of things can be a cofactor. But if this cofactor specifically is non-protein and organic, CH, contain the CH, which is basically vitamins. This will help the enzymes to work. And if you ever wonder, they always tell you like, you need vitamin C, take vitamin D, take this vitamin. You hear about the vitamins. What exactly is the use for the vitamins? Why do we need vitamins? Because it's cofactor to help the enzymes. What if I don't do it? The enzymes are not going to work correctly. So your enzymes are not going to do the job. So you're not going to do the reactions. So you will have an issue, right? Vitamin deficiency. Proteins, if you expose them to certain things, it's going to denaturate. What's denaturate? Remember D, prefix that means no or without. Denaturation, changing the nature, not the nature, not the natural. Like a very simple example, if you get an egg, raw egg, and just open it, you will see the white is semi-solid or semi-liquid, right? It's going to fall, right? Expose it to heat, what's going to happen? It looks completely different. It was transparent, it was clear, and semi-liquid. If you expose it to heat, it will become white, solid, isn't it? We boil the water, we boil eggs, right? So if you expose any protein, whatever it is, any protein, if you expose it to heat or change the pH, example, of course, nobody did that, but I'm telling you, if I get this same raw egg, one egg, and open it, it looks clear, it looks semi-liquid. If I get some acid, battery acid, and put it on it, guess what? Same thing. It's going to become white solid. So it's not only heat. Heat is what we do to eat it. But there are other things, including changing the pH. Denaturation. It's going to lose its nature. Denaturation. Same as lipids, we can add things to the lipids. Here we can add things to the protein, but not for the same reason. For the lipids, we added something in order to make it water soluble, right? Remember this? Phospholipids, glycolipids, are we following? But now we add it for another reason. Here we can add glycoproteins So you're adding glyco. What's glyco? Carbohydrate or sugar. You're adding it to the proteins. How about the proteoglycan? Same thing. Proteoglycan, sugar and protein. Glycoprotein, sugar and protein. What's the difference? There is a difference. What is it? It's the proportions. How much lipids and how much protein and how much carbohydrate? Is it mainly protein and a little bit of carbohydrate, or it's mainly carbohydrate and a little bit of proteins? So basically, the one that's at the end is the main part. Just to remember it, glycoprotein. The name gave it away. It is sugar and protein, isn't it? What's the difference? What does it end with? Protein, right? So the main bulk is protein plus some sugar. Proteoglycan is the opposite. Proteo what? Glycan, like. So which part is the more bulky? Sugar is more bulky, right? And the reason for adding sugar to it in the first place is to make it more viscous, sticky. But you need to know the difference between those two. It's not the same. Both of them are carbohydrate or sugar plus proteins, yes. But which one is more? Last one of the organic molecules or organic compounds is nucleic acid. Nucleic acid um, is what help us store our information, genetic information, and help us to make proteins, synthesize proteins. These are nucleic acid. One of the nucleic acid contains sugar called ribose. What's the ribose? 5 carbon molecule. What do you think if this is ribose is 5 carbon molecule? What do you think about hydrogen and oxygen? How many? I'm telling you, carbon is 5. How many hydrogen? 10. Yes. Remember the ratio? 1 to 2 to 1, isn't it? Yes. So 5 hydrogen. How many hydrogen? 10. And how many oxygen? 5. Yes. So you can expect the ratio without carboxyl at the end. That's it. So ribonucleic acid, what does it mean? It is a nucleic acid that contains sugar called ribose. What's a ribose? 5 carbon sugar. We don't need to know the details. This is what it is. The next one is same thing exactly, but I get this ribose and I take off oxygen from it. What do you call it now? Deoxyribose. Are you guys following these medical terminology, prefixes, suffixes? Ribose, take oxygen. Deoxyribose, right? So this is a nucleic acid also, but is it ribose? No, deoxyribose. And that's why they call it deoxyribonucleic acid, deoxyribo. What's deoxyribo? Ribose, and removed oxygen from it. Very similar, but they are doing different functions. DNA is for inheritance. This is what's carrying our genes to determine what we're going to inherit. That's one. Number two, this is the manager of making proteins, right? So this is DNA will be sitting in the nucleus of the cell. We didn't do the cells, we will do it. But it goes in this nucleus of the cells. I always call the nucleus of the cells the manager's office, okay? It's sitting in the manager there. There is a manuscript telling you how to make protein number 1, 2, 3, 4, all different types of proteins. It's in the DNA. And what protein do you need? I'm not going into the details. What protein do you need? Protein number 1. Okay, can you give us how to do it? Yes, it's this part. Okay, so it contains all menu for all proteins. Okay, and by doing this, by directing protein synthesis, it can also control the enzyme and metabolism since it is responsible for making proteins. How about the RNA? No, this is different. This is not the manager. These are just workers. So who is the manager? DNA. I'm carrying the genes, I'm carrying the instructions for all different types of proteins, and I need RNA as workers to help me. You will do something, you will do something, you will do something. We have different types of RNA that are going to help. So what's the difference in the structure of DNA and RNA? I already told you that the sugar is ribose in RNA, deoxyribose in DNA. And this is where the name came from. DNA, deoxyribo versus ribo. And then there is a phosphate and nitrogenous base in both of them the same. So the comparison here is important. What I'm talking about the structure. They have different structure, they have different function. What's the different structure? The sugar is different. This is ribose, this is deoxyribose. This is number one. Phosphate, it's not different. Both of them are the same. And the third part of this molecule, nucleic acid, is nitrogenous base. What do you mean nitrogenous base? A base that contains nitrogen. What's the difference? There are 4 different types of bases. 4 bases. 3 of them are the same in DNA and RNA. Only one that's different, which is the T. So AGTC, this is DNA. If you take off this T, you replace it with U. This is the only one that's different. So look at this. Here it is. You have sugar, you have phosphate, and you have nitrogenous base. Is this DNA or RNA? You can't tell. You cannot tell. Because how did you know that this sugar is ribose or deoxyribose? You can't tell, right? Phosphate, it's both in the same, in both of them. Nitrogenous base, you don't see any information here. So there is no way to tell. But the bases, generally speaking, are in the form of purine. This is the chemical nature. Again, we don't need to go into the details. Just what's the nature of the bases? It's either purines like adenine and guanine, or it's pyrimidines like the other 3, cytosine, thymine, and uracil. So we use the first letter of each one of those. Adenine is A, guanine is G, as you see here. Cytosine is C, thymine is T, uracil is U. DNA, another difference. We talked about the difference in the structure itself, right? Beside that, what else? The DNA is a double strand, and the D should remind you. DNA, double strand. Look at this. This is a strand. So RNA is like this, just one strand. How about the DNA? Double, like this. They are twisted on each other. How many strands do you see here? 2 strands. Double strand. DNA is double stranded. RNA is single. This is RNA. DNA is different. It looks like this. 2 strands twisted around each other. And can you like untwist it and separate? No, there is a hydrogen bond that keep those together in that shape. So there is one, in the DNA, there is one strand, and the other one is called complementary strand. Okay, this is what we call it. Twisted, and what's keeping them is hydrogen. How about the RNA? The RNA is actually the workers. Workers do different things, right? If you go to any factory, this one is putting things together, and this one's carrying the stuff here, and this one is cleaning, and this one, right? Everyone is different. So we have 3 different ones. Everyone is a worker that's doing something specific. It's messenger RNA, transfer RNA, ribosomal RNA. 3 different types. Each one is doing something specific. And we call it the first letter, M, T, and R, RNA. The way these bases together come together in the DNA is they fit in a specific way. Okay, look at this. If you have this amino acid looking like this, and this amino acid looking like this, they can fit together. If you're having one like this and one like this, you can fit together, right? But you cannot fit this into this, right? What fit with what? ATCG. Memorize it. ATCG. A fits with the T, C fits with the G. Can I put the G and A together? No, you cannot. They look different. ATCG in that specific sequence. If you have A, you have to have T. Otherwise, you won't fit together in the two strands. This is for the DNA, but you said there is one that's different in the RNA, right? Which one is not in the RNA? ATCG. Which one of the four? T. So what do we have instead of the T? U. So what is it? AUCG. This is how they come together. So look at this. Do you think this is DNA or RNA? This one right here. DNA, because you have two strands. Notice this. A, T, C, G, T, A, C, G, C, G, A, T, G, C. It doesn't have to be A, T. A, T, T, A. It's the same. Is it this or is it this? There is no difference. A fits with the T. That's it, period. Which one is here and which one is here? It doesn't matter. It's all about the shape, okay? So this is a double strand, ATCG. If this was RNA, you wouldn't have the T, right? Besides, it's double strand. So this is obviously DNA. And look at this one strand here. What do you think? RNA, for two reasons. Is this a single strand or double? Single. And you see T? You see instead U. So this is RNA. So here is a comparison. I already talked about it. ATCG here versus AUCG. Double strand, single strand, and the function is different. This is good enough to know. What I already told you is good enough. Next is high energy compounds. We have compounds in our body that store and provide us with energy as needed. Okay? You need energy to do everything. Everything. All functions need energy. How to get the energy? From these compounds, high energy compounds. The word phosphorylation, to phosphorylate something, meaning to add phosphorus to it. Okay? Phosphorylate means to add phosphorus to it. So here are the different types. Look at this. Adenosine triphosphate. Translation. And follow this and you'll remember it. What is it again? Adenosine triphosphate. What does it mean? Cut it into pieces as usual. Adenosine. Adenosine. This is adenine. Put sugar, it will become adenosine. Is that clear? S-I-N-E. It means sugar. Adenosine. Adenine sugar. And then what? If you add one phosphate, adenosine monophosphate. Clear? How about two? Adenosine diphosphate. How about three? Adenosine triphosphate. Is that clear? Okay, listen to this. Every time if I wanted to add phosphate, I have to put energy inside. Okay? Here is adenosine. I want to add a phosphate. Put energy first. Put some energy. Here is phosphate. I want to add another one. Put energy first. Put the energy, attach. Try. Put energy, attach. Now I have this structure here, adenosine triphosphate. Is that clear? So now it is storing the energy. I want to use the energy. Yes, just break down one of the phosphate, and what's released? Energy, right? Are we following or not? Following? Break down one of the phosphate, the energy that was here, it will go out, right? Use it. But what did you change adenosine triphosphate to? If I remove this, what is it now? Adenosine diphosphate. Now I have extra energy. I want to store it. Put the phosphate back, put the energy, put the phosphate back. Adenosine triphosphate. I need energy. Remove it, release the energy, and use it. I have extra energy. Return it back. We always go in that ADP, ATP, ADP, ATP. We always change it. Do you need energy? Break down ATP to ADP. Get the energy. You're eating, you're getting a lot of energy, store it back. Okay? The enzyme that help you to do that, adding or removing that phosphate, is called adenosine triphosphate ASE. Just add ASE at the end. Take it as a rule. ASE is a suffix that means enzyme that's doing something. You will see it a lot. Not only this one. Anytime when you see ASE at the end, it's an enzyme that's doing something. Adenosine triphosphate ASE, an enzyme that's helping the adenosine triphosphate, right? Protease, enzyme that work on protein. Carbohydrates, enzyme that work on carbohydrate, and so on. Okay? This is the rule of the thumb. So if you look here, this is adenine, add sugar, ribose. It become adenosine, add phosphate. And then if you wanted to add phosphate more, you have to put high energy bond to store the energy for you. Now we have adenosine diphosphate. I want to add another one. You have to add energy first. Energy in the form of what? It's called high energy bond. I told you this is adenosine. I want to add one phosphate. Put energy, right? In the form of what? High energy bond. Okay? The energy that's stored in between is called high energy bond. And that's it for this chapter.